The gas chromatography, mass spectrometry or GCMS, is one of the so-called hyphenated analytical techniques. It is actually two techniques that are combined to form a single method for analyzing mixtures of organic chemicals. Gas chromatography separates the components of a mixture, and mass spectrometry characterizes each of the components individually. The combination of the two techniques allows for both qualitative and quantitative evaluations of a sample containing a number of organic compounds. GCMS can be used to study liquid, gaseous, or solid samples. The GCMS is used in a variety of different disciplines in chemical, astrochemistry, medicine, geochemical, industrial, security, law enforcement, food, beverage and perfume analysis, sports anti-doping analysis, pharmaceutical, biological, geological, environmental, and forensic research. Where a chemical analyzer is used to detect chemical compounds of interest or to identify the different components of the unknown mixtures, let us say for example drugs, poisons, toxic chemicals, gunshot residues, explosives, and even material samples obtained from the planet Mars. Some of the applications are identifying and quantifying unknown samples, identifying unknown contaminants, environmental analysis, identifying trace elements in samples, identifying gases in a sealed environment, and identifying residual solvents. To perform these analytical techniques in a laboratory, a GCMS instrument is being used. It is composed of two major building blocks, the gas chromatograph and the mass spectrometer. But the question is, how does this GCMS works inside the laboratory? Analysis begins with the gas chromatograph to separate a sample into its individual chemical components. A chemist gathers available samples, and then a sample of the analyte is introduced by a syringe injection into the heated injector tube, where it is vaporized and mixed with the carrier gas,
where the sample is effectively vaporized into the gas phase and separated into its various components using a capillary column coated with stationary phase. The compounds are propelled by an inert carrier gas such as helium, hydrogen, or nitrogen. As components of the mixtures are separated, each compound elutes from the column at different time based on its boiling point and polarity. The time of elution is referred to as compounds retention time. Once the components leaves the GC column, they are ionized and fragmented by the mass spectrometer using electron or chemical ionization sources. Ionized molecules and fragments are then accelerated through the instrument's mass analyzer, which quiet often in a quadrupole or ion trap. It is here that ions are separated based on their different mass to charge ratios. GCMS data acquisition can be performed in either full scan mode to cover either a wide range of MZ ratios or selected ion monitoring mode to gather data for specific masses of interest. The final steps of the process involve ion detection and analysis, with the fragmented ions appearing as a function of their MZ ratios. Peak areas, meanwhile, are proportional to the quantity of the corresponding compound. When a complex sample is separated by GCMS, it will produce many different peaks in the gas chromatogram, and each peak generates a unique mass spectrum used for compound identification.
Using extensive commercially available libraries of mass spectra, unknown compounds and target analyzed can be identified and quantified. This video narrative is submitted in partial fulfillment for the subject Chemical Biology 2 or Analytical Methods for Biology. We are first-year BIS biology students at Bukidnon State University. We would like to thank you for listening and watching our narrative video together with my partner in this output, Johny Alegarbes, and yours truly, Annalie also. We hope that you too learn and enjoy our video.